You'll find these notes in Chapter 14, Section 1 of your textbook, and it is a small subset of a section that involves the chromosomes, but toward the end of the section, it talks about traits that are found on the sex chromosomes, which we are going to call sex-linked traits. So we are going to be focusing our attention on the X and the Y chromosomes with regards to sex-linked traits. As a reminder, humans have 46 chromosomes, and they are in 23 pairs, and the last pair are called the sex chromosomes. They determine an individual's genetic sex. The remaining 44 chromosomes are called autosomes. And autosomes do not influence whether you are genetically male or female. They code for traits that involve you being just a human being. You know, the fact that you have amylase as an enzyme, or the fact that you make keratin in your hair has nothing to do with your genetic male or femaleness. Now, a clarification of the words sex versus gender. Genetic sex is a label, male or female, assigned by a doctor at birth based on the chromosomes you have and external genitalia. So when the doctor holds you up and you were born, they say, oh, we have a boy, or oh, we have a girl. Gender is much more complex, and we typically, inherently want to use these words interchangeably. However, they are not interchangeable. They are actually different concepts. Gender is a social and legal status. It's a set of expectations from society about behaviors, characteristics, and thoughts. Gender has variability within cultures and even time periods. So a really easy to understand example is masculinity through the ages and through clothing. Oops. So these are depictions of male dress in the 1600s, and I think even going up to the 1700s. You had long hair and very flowy collars, skirts, pantaloons, and this was considered to be very masculine. But, in America today, this is probably not a typical dress for a man. So you can see that what was a cultural and time period standard for what masculinity was has shifted through the ages. So gender, instead of being about DNA and body parts, is more about how you're expected to act because of your sex. And then finally, gender identity is how you express your gender through clothing, behavior, appearance, etc. Now, I wanted to touch upon those concepts because with regards to our class and these notes, we are going to be discussing genetic sex for the sake of the flow of chromosomes from parent to offspring. So it's important to note the difference between genetic sex and gender, but for the sake of biology class, we're going to be focusing our attention on genetic sex to discuss inheritance. So let's go back to chromosomes, and I want to just reiterate, we've talked about this earlier, that men have X and Y as their sex chromosomes, and you can see those here. The Y chromosome, you can see, is much smaller than the X chromosome. So this would be a karyotype. Of a genetic male. And women have XX as their sex chromosomes, and the two X chromosomes are the same size. And those are here. Now, if you're a female, all human egg cells, so the gametes produced by genetic females, will have one X chromosome. So essentially, all eggs are going to be passing an X to their offspring. So women just make eggs with X chromosomes, because women can only give X to their children because that's all that they have. They do not have a Y chromosome. Now, with regards to men, half of the sperm that men produce will have an X and the other half will have a Y, again, because of the separation of chromosomes in meiosis. And that is why there is a 50-50% chance for a male or boy or a female, genetic female, offspring with every pregnancy. The process that ensures that gametes only have one sex chromosome is, as you know, meiosis. This Punnett square shows how women can only produce eggs that have X chromosomes, and men can produce half of their sperm with an X and half of their sperm with a Y. So depending upon what sperm fertilizes the egg, there is a 50% chance of having a genetic girl and a 50% chance of having a genetic boy with each pregnancy. There's a misconception that if you have, let's say, 3 girls, you're more likely to have a girl with the 4th pregnancy. And that is not true. Every pregnancy is an equal 50-50% chance, and it's just like flipping a coin. One flip has no influence on flips that come later, just like one pregnancy does not have any influence on the sperm that fertilizes an egg later. Now let's talk about the sex chromosomes in a little bit more detail. Because the X and the Y chromosomes determine genetic sex, the genes on them are called sex-linked genes. The Y chromosome has approximately 140 genes on it, and most of them are associated with male sex determination and sperm development. The one with the most importance is called the testes determining factor. And the presence of that gene initiates the formation of what are called seminiferous tubules and in the male testes. And in the seminiferous tubules is where the meiosis of sperm occurs. The Y chromosome is only passed from father to son. The X chromosome has more than 1,200 genes on it, so it is much larger. And not all of those genes are women specific. Males have to have an X chromosome just like females have to have an X chromosome. Now, because it has many more genes on it, there are more chances of a mutation occurring on the genes. And a mutation is a change in the DNA that can cause a genetic disorder. Some of the mutations on the X chromosome can be serious, or some can be mild, some can be serious, and they can cause some genetic disorders, some of which are outlined on this table here. Examples that many students are familiar with are color blindness and hemophilia, which is a bleeding disorder. Muscular dystrophy, Duchenne's muscular dystrophy, is also another example of a mutation found on the X chromosome. So examples are color blindness, ALD, hemophilia, and muscular dystrophy. Most X-linked chromosomal disorders are recessive, meaning you need 2 copies in order to get the disorder. So for a woman, she would need 2 copies of a mutated gene for hemophilia to exhibit hemophilia. But men only have 1 X. And so what does that mean for them? Women can be heterozygous for X-linked disorders and not show the disease. But since men only have 1 X chromosome, they will always show the disease because they don't have another X chromosome gene to mask a mutated or defective one. Women will need to get 2 defective X genes in order to show an X-linked disorder. Men only need 1 copy. So the easiest way to understand this is kind of looking at a diagram. If a male, the father, does not have an X-linked disorder on his X chromosome, and he marries a woman that does, so let's say this is, this little r we're going to say is color blindness. She's not going to show the color blindness trait because she has 1 normal X chromosome and 1 that is mutated that does have the color blindness gene. But the normal X chromosome will mask the defective colorblind gene. What happens is we're going to call her a very specific name, and that's called a carrier, because she's carrying the color blindness gene, but not actually showing the color blindness trait. So I'm going to highlight that. Carrier is a new vocabulary word for you. And if she marries a genetically normal, no color blindness male, this is kind of like a Punnett square. So it shows all the combinations of the X and the Y chromosome from this father and this mother. If the father donates a normal X and the mother donates her normal X, they're going to have a completely non-affected daughter. If the father donates a normal X and the mother donates a abnormal X, they would have a carrier daughter. Now, if the father donates a Y chromosome and the mother donates her normal X, they'll have a non-affected son. But lastly, if the father donates a Y and the mother donates a color blindness X, they will have an affected son. So in doing this cross, you'd see that the only affected child is a son who is going to be colorblind. So males, have a much higher chance of getting X-linked disorders compared to females, because with only 1 X, that has the mutation, they will show the disorder. So lastly, I want to show you how to do a Punnett square that involves sex-linked traits. To do these Punnett squares, you are now going to be listing the X and the Y chromosome when you do the cross. So in order to have genetic offspring, you have to cross a genetic male with a genetic female. And so you will always set up your Punnett squares that way. And this one got a little cut off. Sorry about that. I'll fix it. So the male's, the male's X and Y chromosome crossed with a female's XX chromosome. And we are going to put the gene that is affected on the X chromosome. Do not put the gene on the Y. So for example, with color blindness, normal vision is dominant, color blindness is recessive. We're going to cross a carrier female with a normal male. So our normal visioned male is going to be X, big B, Y, and our carrier female is going to be X, big B, X, little b, and we are writing these as a superscript, similar to how we noted the alleles for codominance. So same as before, bring over and bring down. And for these problems, I just want you to worry about doing your phenotypic percents, not your genotype listing. You can just do phenotype. And I often will separate out male and female. So for our females, phenotypically, we're going to have 100% normal vision. Yes, I know we have a carrier here, but phenotypically, she's going to be normal. For our males, we are going to have 50% normal vision and 50% colorblind. Now you might be wondering, well, wait a minute, Crosby, how did you get 50% when you're looking at one box? Isn't that 25%? And the answer is if you separate the boxes by male versus female, you now have boxes that are 50% each, not 25% each. So 2 boxes for females is going to equal 100, and 2 boxes for males is going to equal 100. The problem below is crossing a colorblind female with a normal male. So a colorblind female is going to be X little b, and a normal visioned male is going to be X big B, Y. Again, remember, do not put a gene on the Y chromosome. You're also going to make sure you put the capital B before the lowercase b. We're just going to do phenotype percentages. We're going to break it down by female offspring and male offspring. And in this case, all of our daughters are going to be normal vision. And all of our sons are going to be colorblind. If a mother is colorblind, all of her sons are going to be colorblind because she will always pass a colorblind X to her son because the Y is coming from the father. And that's the only allele she has to give. So colorblind mothers will have colorblind sons. So just remember, you must use the X and the Y in the Punnett square, and you have to have your allele as a superscript. Do not put the gene on the Y chromosome.